Hi, everyone. Welcome, and thank you for joining us for this final day of the 2022 annual meeting of the California Oak Mortality Task Force. If you were here with us before, you've heard these same kind of housekeeping. I just want to tell you that today's session is dedicated to the work of the Phytophthora and Native Habitats Work Group and is focused on Phytophthora and restoration areas and natural areas. Our moderator today is Janelle Hillman of the Santa Clara Valley Water District. So as in the past 2 days, each of the speakers has been allotted a few minutes to answer any questions at the end of their presentation. Everyone should be muted. So if you have a question, please use the chat to type it in, and Janelle and I will facilitate getting those questions answered during the speaker's time. Any questions that we can't get to during the actual speaker's time, we will save to the end of this session, or we'll follow up with you offline later. The session is being recorded and will be posted online at the suddenoakdeath.org and the calphytos.org websites. And with that, I will hand the meeting over officially to Janelle.
Good afternoon, everyone. Thanks so much for joining us on day 3 here of the California Oak Mortality Task Force annual meeting and our own annual meeting for the CalPhytos Working Group. I just want to start with a special thank you to Janice Alexander and Susan Frankel for all their work with the CalPhytos group and working to organize today's panel of speakers and a whole series of really interesting and diverse talks that we have today. So I'm really honored to have Sarah Green join us as our first speaker today. Sarah Green is a researcher for UK Forest Research in Scotland. Her current projects include early detection of Phytophthora in nurseries and traded plants in EU and third countries, which she will present today. This project is an international collaboration involving 10 partner countries and aims to develop a coordinated strategy for the early detection of Phytophthora pathogens in plant nurseries and traded plants for planting across EU and third countries in order to inform best practice. Sarah also has a whole suite of other really interesting projects, including molecular detection of Phytophthora in forest, woodland, and urban garden environments using an Illumina metabarcoding technique, and evidence for natural resistance in Japanese larch to Phytophthora ramorum and in juniper to Phytophthora austrocedri. Please welcome Sarah.
All right, thank you very much. Obviously, I'll share my screen. Thank you also for inviting me to speak today. Just get this up. I haven't been able to attend the sort of meetings so far this week because I've been down at Alice Holt in the south of England at a tree health meeting, which I got back from quite late last night. So that's why I haven't been able to attend so far. Anyway, I will be staying on for the couple of talks after mine, but then it will be getting quite late. So, right. I think you should be seeing the full screen now. Is that correct? Okay, so I shall start. Yeah, so I as per the introduction, I'm going to be talking about some of the work we're doing on Phytophthora detection in plant nurseries and how we're trying to support best practice. Just would like to acknowledge that there's a whole team of people involved in this work across a number of institutes, and I won't go into the names now because I'm sort of short of time, but yeah, you'll know some familiar names, for example, David Cook, and then maybe Louise Barwell and Beth Peirce, who have been sort of very instrumental in the modelling side of things. So as a forest pathologist, I'm quite interested in Phytophthora just because of the number of invasive Phytophthora we have been dealing with in the UK over the last 20 years. You'll be familiar with most of these, I think. And the most recent outbreak has been Phytophthora pluvialis on western hemlock and Douglas fir, and that was discovered last year. And with all of these Phytophthora, we know there's a very strong link to their introduction and spread as a result of being transmitted in the plant trade. And so because of this, we've had 4 projects really over the last, well, since 2016, which have been looking at the spread of Phytophthora in trade and Phytophthora diversity in relation to management practice and other sort of practices as well. So the main one, which I'm going to present results from today, is the Phytothreats project. And Susan Frankel will be quite familiar with that because she came over and spoke at one of our first meetings, and I was able to show her around a little bit as well. And that project ran from 2016 to 2019, and we developed the methods that I'm going to describe during that project. And then, as Janelle said in the introduction, the methods that we developed for detecting Phytophthora in the nurseries using metabarcoding were then rolled out in a wider project involving 12 partner countries, which was the ID Fight project, which started in 2020. And 6 of those partner countries have been able to actually get out and sample nurseries using the methods that we've developed. But most of the results I'll be sort of talking about today really are coming from the Phytothreats project, which is the one outlined in bold, because we are still processing samples from the ID Fight project. And then there are 2 other projects, much smaller projects, which you may well be interested in, but I'm not going to present results from. But since given what your interests are, we've had a small project where we've been looking at Phytophthora risks in nursery waste piles, because we found that all nurseries really struggled to know what to do with their waste, with their spent growing media and their green waste, and largely it just gets dumped at the edge of the nursery and often next to streams and hedgerows and woodland. And we have been finding and baiting Phytophthora from these waste piles, including Phytophthora morum. And I think just because of the nature of where they're located and the fact you get a real mix of Phytophthora sort of dumped in there, they do present quite a hazard and can result in provide a conduit in a way to Phytophthora spreading from the waste into the wider environment. So this is something we have been working with, also with the certification scheme, which I'll talk a little bit about at the end. And then the other sort of issue of concern that we have a bit at the moment is the move to peat-free growing media, which is good from an environmental perspective, because peat is a finite resource. But the banning or the phasing out of peat in the particularly in the amateur market is raising some concerns about what the other constituents are and how they're sourced and how they're processed. And some of these constituents include chopped bark, wood fibre, processed green waste from sort of urban areas, and also coir imported from the tropics. So yeah, these are just some of the projects we've been working on. Hang on, next slide. Well, here we go. So the aim of the talk today really is just to give a very brief overview of some of our research findings to date, hopefully highlighting some of the findings that may be of most interest to you, and also how our science and our working with nurseries is perhaps helping to support best practice in the UK and in other countries as well. So I'll start off by talking about how we did our analyses of Phytophthora communities and nurseries. So we were really lucky to be able to have a good number of nurseries to work with who offered themselves up for sampling. And it wasn't really that much of a struggle, to be honest, because I think a lot of nursery managers really do want to improve practice, and they were quite keen to be engaged in the science projects. They were all anonymized, and so then they're not named in any reporting, so which kind of helped in getting them on board. So in the UK, we've been working with 17 different partner nurseries operating a range of practices and located right across the UK. And then we've got these 6 other partner countries who have been sampling in their own countries as well. And that includes Greece, Ireland, USA, and California, and also Italy, France, and Austria. Anyway, so at each of the nurseries, we've been collecting water and root samples in triplicate, and we're collecting also all the data on the sample, for example, the host species, where it's come from, location on the nursery, and when it's water samples, what type of water sample. So each nursery was also subject to a questionnaire so that we could list some of the key management practices, and that will enable the analysis to be done in relation to management practice and Phytophthora diversity found at each nursery. And we've tested a range of plant hosts. It really just depends on what's there at the nursery and what the nursery manager wants us to look at. And then we are analysing the samples for Phytophthora using a DNA metabarcoding approach, which I'll outline briefly in a minute. But for our more recent projects, we've also been adding baiting to get Phytophthora into live culture and improve viability. As the projects have progressed, we are regularly feeding results back to the nursery managers, along with recommendations along the way on how to improve management. Okay, so we found these Phytophthora in your nursery. What does it mean? And how can you try and mitigate that? And then also our aim is to work with the accreditation scheme that's developing in the UK at the moment, and also in other countries that are partners on our IDFAC project, and to develop best practice guidance and support accreditation initiatives. So in terms of the water sampling, we go to the nursery, we sample all the water that they use for irrigation, whatever source of water they have. We also sample from drainage, ditches, ponds and puddles around the nursery, rainwater collection butts and any streams that are running alongside nurseries. And the water is collected into a knapsack sprayer and it's pumped through a cellulose filter. I hope you can see my cursor here. And the Phytophthora propagules basically are collected on the filter, and that filter gets put into a buffer solution and the propagules wash off, and then we do the DNA extraction from the buffer. What we also do is to take batches of plants, sit them into trays, water them thoroughly so that the water runs through the pots, runs through the soil and the roots and collects in the bottom of the tray. And that washes through any Phytophthora propagules into the water, and that's pumped through as well and tested. And then we also sample roots from the water flow batches and from other batches of plants as we see fit. You know, there's a mix of symptomatic and asymptomatic hosts. And then as I talked about the problem with the plant dumps, and we also sample roots from plant dumps as well. And then this is a slide which provided by David Cook from the James Hutton Institute. David's been a close partner on all the projects that I'm talking about now, and developing the methodology. So the samples that we test are then root samples, which are freeze-dried, and then the filter, which is sitting in a buffer solution. And the DNA is extracted from the buffer and the roots and subjected to a PCR, which is a nested PCR, which was published in 2012, again by David Cook and team, and it targets the ITS1 region. And then there's various steps which I won't get into, which prepare for Illumina, which is high throughput sequencing, and then it's sequenced at the James Hutton Institute on an Illumina MySeq. So then what we get is a load of sequence data, and actually over the last few years there's been the development of this Phytophthora pipeline to classify the sequences, to identify them to a species level, and this was also developed at the James Hutton Institute by David Cook and again Peter Koch, Leighton Pritchard and Peter Thorpe, and that's also publicly available, as you can see here online. And because we're working with nurseries, the pipeline is very much focused on maximizing accuracy and minimizing error. So it's minimizing the risk of false positives, but as a result, we're likely to get more false negatives. There's a lot of quality control and cleaning that goes on. We do use what we call synthetic DNA sequence controls on our plates. So that acts as a check for cross-contamination across samples on a plate, but it also acts for a check for PCR error and Illumina sequence error. And then, so a lot of sequences are thrown out if they're deemed to be error, and then any remaining sequence is then aligned to a highly stringent reference database to allow that sequence to be identified to a Phytophthora species. And David Cook and colleagues developed that pipeline, that database manually. And so that's been used on all our projects. But as I said, for the more recent projects, we've also been baiting for Phytophthora, where we take the water samples and we test to see if Phytophthora is present by floating bait leaves on top, or in some cases, some countries like the US and Italy like to put pears in. And so this works on the principle that Phytophthora has free swimming zoospores that can swim to the bait leaf, and then the lesions develop, they're plated onto selective media, you do colony PCR and sequencing of the live cultures to identify them to a species level. And again, having those live cultures growing out does prove that that species is viable and present. So now I'm going to move on just to outline again, quite briefly, but pick out some key results from some of the modelling analyses that were done from the data that's arisen from the Phytothreats project. And this was done by, this work's been done by Louise Barwell with Beth Peirce, who are collaborators at this institute, CEH Institute in the UK. And so the pipeline produces data in the form of an Excel file, which lists each sample, all the data associated with that sample, and all the Phytophthora species associated with that sample as well. And also data on whether the samples that were negative for Phytophthora too. So we're getting everything. So they were able to take Phytophthora data. So this is still a work in progress, and more data are being added from more nurseries, including from other countries. But what I'm going to present at the moment is just the data that's complete for the UK nurseries. So this is data from 1,000 samples from 16 UK nurseries, and this was around 600 plant samples and about 400 water samples. And then also included in the modelling analysis is a database of about 19,000 UK Phytophthora records, not just from nurseries, but from wider environments as well. So we can start to look at what's appearing in nurseries in relation to what's also being found in the wider environment in the UK. And then because there's an interest in what Phytophthora traits, their biological and ecological traits, and how that might affect their ability to spread and establish. A database of Phytophthora traits has been developed by Louise and Beth, but also in collaboration with teams in Australia and New Zealand. And so these were the inputs that went into the models. So again, sorry, it's a bit hard to read, but I want to show this, and hopefully you can pick out. This is basically the key Phytophthora species and the key host species that went into these particular models. And again, these will be refined, and we hope to add more species as the models are developed. But so far, what you can see in this figure A is a list showing the most abundant Phytophtheras that we found across all the 60 nurseries included in this analysis. And so for those of you who are familiar with the different species, you can see, probably in some cases the usual suspects, but the ones in bold are the Phytophtheras that were actually included in the models, because these were Phytophtheras that could be identified to a clear single species, whereas the ITS1 region in some cases doesn't allow differentiation between two or more species, and up until now those haven't been included in the model, but we hope to again be able to change that and include more species in the models. And when it comes to the hosts, you can see the most frequent host species that come into the models are juniper, yew, cherry, oak, and Chamaecyparis, and then we get to some of the ornamentals like rhododendrons, camellias, and hebes and lavenders. And again, the hosts outlined in bold are those that were included in the models and the most abundant sampled across the nurseries. So just one of the aspects that we were quite interested in was what host species, what plant species had the most Phytophtheras associated with them and therefore could be considered to be highest risk. And this was a bit surprising because we didn't expect that the most sort of hot, sort of risky plant species was in fact Pseudotsuga. So that's Douglas fir, was found to have pretty much some of the highest number of Phytophthora species associated with it, including Phytophthora pseudocryptogeus and amomi, plurivora, and syringae, for example. And this is of interest in the UK because Douglas fir is one of those species which is going to be planted much more widely and is touted as being one of the more important forestry species going forward into the future, particularly with climate change. So really, we don't want to be spreading more Phytophtheras into the wider environment through planting trees that have all these Phytophtheras associated with their roots. So this is a clear message we're going to be passing on to forest nursery managers about, raising plants that are actually not containing all these associates of Phytophthora in their roots that are just going to be transferred to the wider environment. And other high-risk hosts that we found were Chamaecyparis and then Fagus, Prunus, again, yew, hebe, and then some of these ornamentals. But it was a little surprise to us that we found so many. It was some of the forestry tree species that were the most high risk in terms of Phytophthora associations. And then the other area of interest was the effect of sample type. So this graph is showing which Phytophthora species are most associated with water samples versus those which are most associated with root samples, and those which are equally associated with root or water samples. So those in blue are those which are significantly more associated with water samples. And again, we have some of the usual suspects, the clade 6 species like Phytophthora gonopodioides, for example, that is common in water. But then there are other Phytophtheras that was a bit of a surprise to us, that they were almost always found in water, and that included Phytophthora pseudosyringae, Gallica, and Hibernalis. So that was a slight surprise. And then there are the others that in green here, which are most likely to be found in roots, and those include Phytophthora cinnamomi, Cryptogyra, Pseudocryptogyra, and in Cambivora, for example. But what is actually probably of most interest is these ones in grey in the middle, because they're equally likely to be found in water or root samples. So therefore, they would have perhaps a wider ability to spread. Their lifestyles allow them to spread equally in water or equally in the actual host. And those do include some of the more important quarantine-regulated species. For example, Phytophthora ramorum, Phytophthora ostracegae, which is killing juniper in the UK, and then others which are known associated with certain plants in the trade, like Phytophthora occultans, Phytophthora pseudotsugae, which was actually a first record in the UK. So we thought that was actually potentially quite interesting, allowing us to understand a bit more about how these Phytophtheras might be spreading.
Sarah,
and so
you've got about 2 more minutes, just so you know.
Okay, so I have probably overrunning my time a little bit, so I will move on quickly. So then we wanted to look at the effective management practice, and so the modelers put together a list of best practice criteria that we were able to obtain for all nurseries, but that also showed variation among nurseries. So that also included size, their latitude, turnover, what they spend on buying in plants, but also irrigation sources. Do they use open or closed water sources? Do they treat water or not? Do they use their own growing media or commercial mix? Do they reuse pots or not? Raised benches, disinfectant stations. Do they import whole plants or not? Do they have a quarantine holding area? How do they dispose of their plants? So those were some of the key management practices that were included in the models. And then just an overview of basically the highest risk nurseries were those smaller, lower turnover ornamental nurseries that use open, untreated water supplies. They import live plants, and they tended to have more diverse Phytophthora communities. It consists of more recent invasive species and also rare species not widespread in the wider environment. Interestingly, the commercial tree nurseries had a higher overall probability of detecting Phytophthora, but their communities tended to include less diverse and fewer rare or invasive species. And so finally, I just want to talk about the effect of project engagement. And this graph shows those the nurseries that we sampled here down, which were anonymized, and whether they over a 3-year sampling period, did we find fewer Phytophthora positive samples? And for the nurseries highlighted in blue, yes, we did. As the project progressed, they got cleaner when we found fewer Phytophthora positive samples. And so they were able to change practice as the project went on, including raising plants off the ground, ceasing trade in high-risk hosts such as juniper, improving drainage, employing plant health specialists. One nursery actually installed a borehole to extract water at great cost. And then quite a few of our partner nurseries are joining the plant healthy certification scheme. And this is a scheme that has recently, I realize now I'm now slightly over time, but this is a scheme that you can ask me questions about this. I don't run the scheme, but it started in 2020 being rolled out. They've got about 25 nurseries now joined and about 20 more who are in the process of joining. It's based on a management standard and of risk assessment. So then, and the nurseries are audited according to a management standard, which is still under development. And we're helping to support that management standard by producing guidance on best practice for the horticultural sector, such as this two-sided flyer here, which is part of the standard. And we've also been working with the Plant Healthy Scheme to focus on specific problem areas, such as how you would best dispose of plant waste and spent growing media through recommendations on either composting on site properly or having your waste removed to a commercial waste or composting facility. So I'm going to leave it there. So sorry for running over a little bit. Yeah, this is one of our partner nurseries with their Plant Healthy certification. So ending on a good note. Anyway, thank you very much for listening. And if I've got a minute or two for questions, it would be good to answer some. Thank you.
Thanks so much, Sarah. I think I'm seeing
Sarah.
one question coming in from. Yeah, we have plenty of time for questions for Sarah. So the first question is from Tyler Bure. Tyler says, Sarah, I don't remember seeing P. crassamura listed in your important nursery species, but I did see P. megasperma. Is the apparent lack of P. crassamura methodological, or is it not much of a problem as P. megasperma in the UK?
So those have recently, they cannot be separated now by ITS1. So if you see megasperma now, then it is usually associated or could be crassamura. So we can't distinguish between the two. But I think that might have been added to the database more recently than the data that I've shown you. So does that answer your question as to why it was megasperma on its own and not megasperma / crassamura? Absolutely. Yeah, okay.
And then I actually have a question, Sarah. You mentioned your Plant Healthy certification program, that you've had 25 nurseries join so far. That's really exciting because we're working on a similar certification program here in California that we'll hear about a little bit later today. I'm curious if that program, is that so far just based in the EU, or does that extend to your partner countries as well?
So at the moment it's a UK only initiative, and it's being managed by someone called Alistair Yeomans, based in the UK, very much UK initiative. But so the IDFITE project that you mentioned, that we've been involved with, we can't kind of enforce an accreditation scheme on other people, but I think we're trying to put across what we're doing in the UK and actually trying to share the best practice, for example, the guidance we've put together, and that will be translated into the different partner country languages and disseminated across the key stakeholders that we've identified as part of the IDFITE project. So, but that is still very much a work in progress, and because the project doesn't finish until March. So I'm hoping in sort of a year's time, we'll be able to probably give you more information on how that element of the project, so the more international side of things has developed.
Great, thank you. We have a few more questions. Janice, I'm assuming we can take the questions since we're not sure how long Sarah can stay through the end of.
Yeah,
okay.
Let's get all of her questions done now.
Great, okay. So Sarah, the next question is from Faith Campbell, and Faith asks, the US nursery industry has voluntary BMP program sank, but after 20 years, still only about 20 nurseries participating. Can you work with Craig Real Bruges to try and find ways to improve engagement? Actually, that's a question for everyone, so maybe that's just a comment.
Yeah, so I should make the point that I'm obviously a researcher with Forest Research, and the actual scheme is being run by someone called Alistair Yeomans, and Alistair's always happy to engage with people in other countries about how the scheme is being developed, and also very much aware that the scheme itself, the management standard, does need improving. It's got a fairly low baseline at the moment because the baseline is very low generally across the UK, but it's going to be a gradual process of improvement. Now, can I just say one more thing, that one of the reasons why there's probably been quite a sharp increase in nurseries joining the scheme, particularly over the last few months, is that the government has decided that any new plantings, tree plantings, to be done on government-owned land, publicly owned land, the stock has to be supplied by certified nurseries. So that's, I think it's a phase that is sort of under development at the moment, or it's a trial phase anyway. But I think that has stimulated more and more interest in joining accreditation schemes. There was some comment about cost. There will be a cost for registration as part of the Plant Healthy, but there is a website which is on my presentation if you're sharing that. And if you Google Plant Healthy Certification Scheme, all the information is on there as well. And you can contact Alistair Yeomans directly if you have any questions in relation specifically to that scheme.
Yes, so you did answer, there was a question about cost. Susan is asking if you could just say a bit more about requirements for accreditation, if it involves testing or if it's self-certified.
So no, it involves, it is an audit process. It's not, at the moment there's no diagnostic testing as requirement as part of being part of the scheme, but there's an audit process. So trained auditors will go to the nursery and run through the whole what they call is like an appropriate level of risk. So each nursery is carefully risk assessed through every stage in their process of bringing plants in through to passing plants out. So yes, there is a very clear audit process. I can see someone was asking about who Alistair Yeomans is, and someone's popped in the Plant Healthy certification link in the chat there. That Susan has, yeah. So that's, yeah. So Alistair Yeomans or that website will provide probably much better information than I can. But we've been working very closely with Alistair and trying to highlight the problem areas that we need to work on with nursery managers, like the plant waste issue, for example.
And then just the final question, do all the participating nurseries get a certificate, or is it just the nurseries that don't test positive?
So as I said, there at the moment there is no so our Phytophthora sampling that we did was really for research purposes and to understand what practices are key to be targeted and what were problematic in terms of spreading Phytophthora. For the accreditation scheme or the certification scheme itself, at the moment there is no requirement for diagnostic testing. It's just a check of your site and that you're doing things properly. Each nursery is certified, but that is renewed every year following audit, and then they can obviously state that they are certified on their website and on their but I don't think they're allowed to put it on their products, just because of the supply chains in the UK, that you can still import material but be Plant Healthy certified. It's not necessarily the case that plant would have been Plant Healthy certified for its entire lifespan. So I don't think they're allowed to put it on in the pots, but they can make a statement on their website and generally that they are certified. It's still a process, yeah. Development. So
thank you. Thanks so much, Susan, for the presentation today, Sarah.
You're welcome, and thanks for inviting me. And I will stay on for a bit longer, probably not to the questions at the very end. I'll be asleep by then, I think.
Excellent, yeah. Thanks. Okay, so our next speaker is Jerry Weiland, who is a research plant pathologist with the USDA Agricultural Research Service out of Corvallis, Oregon. Jerry conducts research on plant pathogens affecting trees, shrubs, and vines of the small fruit and nursery industry for the USDA Agricultural Research Service. He's published numerous papers on Phytophthora and Pythium in the Pacific Northwest, and today he'll share some of his findings on pathogen presence and impact in horticultural nurseries. So thanks so much, Jerry, for being here today.
Yes, and thank you for the invite. I'm hoping everybody can hear me. Thumbs up. Yes. So the research I'm presenting today comes from an ornamental nurseries perspective, Most of our funding has come from the USDA itself, or the Floriculture and Nursery Research Initiative, and some from the Horticultural Research Institute. And I was thinking about a subtitle as I was working through this, and I think a really good point that I want to make throughout all of this that I hope you see the storyline is why it is important to know which pathogens you are dealing with. And I'm going to start out with a really brief history of root rot. Root rot of rhododendron is probably the longest, most well-studied root disease of nursery industries. This was first found in 1920s from New Jersey, and disease losses at that time averaged anywhere from 10 to 25%, and that's been about the same sort of losses we've seen in rhododendrons since then. And so really rhododendron sort of serves as the model where we have the most information about Phytophthora root rot in ornamental nurseries. The disease has been primarily caused by Phytophthora cinnamomi. That comes from surveys mainly from the 1970s, 80s, and 90s. So there have been a couple surveys after that that also pinpointed cinnamomi as the primary cause. And basically, there's been 100 years of research on how to control Phytophthora cinnamomi. This includes research on fungicides, biocontrol, compost amendments, host resistance, numerous other disease management strategies. And to date, probably the industry standard are two systemic fungicides, mefenoxam and phosphorus acid. And these are generally considered in the nursery industry here as probably two of the most effective preventative nursery treatments. And you can see some of that historical research off to the right. So in 2013, when I started working on Phytophthora root rot of rhododendrons, I talked to several rhododendron growers, and they were still having problems controlling root rot. So we decided to take a step back and to figure out, well, after 100 years of research, or almost 100 years of research, are we still seeing the same amount of damage? I would hope we would be seeing less with all of these management strategies that have been developed. And the second major purpose of the study was to identify the Phytophthora and Pythium species that were associated with root rot now. Maybe there's been a change away from cinnamomi. Maybe there's something else. And to do that, we looked at rhododendrons in 3 of the production systems. We looked at recently rooted, very young rhododendrons in propagation systems. We looked at rhododendrons with root rot in container systems, and we also looked at them in the field. And we studied plants from 3 nurseries for each of these 3 types of systems. And what we see is that in propagation, there really wasn't a lot of root rot at all. Only about 0.2% of the samples that we looked at actually had root rot or symptoms of disease. 99.8% of what we looked at is healthy. They were healthy, green. We didn't see any problems there. Compare that to the container, where we found about 14% of the crop in container systems. Again, that's 1 gallon size on up. About 14% of the plants we surveyed had root rot, and about 16% of field-grown rhododendrons had root rot. That 16% is a little bit misleading because I've since surveyed other nurseries, and looking at the field systems, disease in field systems tends to run actually higher. I would say the average is more around 25 to 30%. Some nurseries run as high as 40 to 50% of their crop of being affected by Phytophthora root rot. So really, across those 3 systems, the worst is the field system. So this tells me something. Losses due to root rot are still running 10 to 25%. So I would have suspected after 100 years of research that we'd be doing better, and we're not. So why is root rot increasing at each stage? I'd say the number 1 reason for that is contact with contaminated field soil. Again and again and again, you can see in that center picture, there's a container system with some pretty heavy contact between the container plants and field soil, and of course the field system on the right. The one propagation system that we saw that had a problem with root rot was this one on the left, where I suspect some contaminated material got into that bed that you see. And that media there had been reused for a number of decades. So they basically increased their risk there by reusing that media over and over and over. The cuttings themselves are pretty low risk for introducing Phytophthora root rot just because they're taken above ground and they're not typically contaminated. If we take a look at pathogens and propagation systems, 98% of what we recovered from propagation plants were Pythium. And a lot of that is because those were mostly, remember in this case, all healthy plants. Container plants, we see a big uptick in the amount of Phytophthora that we detected. In both container and field systems, we are looking here at primarily plants with symptoms of root rot. And then once we move to the field, we again see an upsurge in the number of samples that came back positive for Phytophthora, 53%. And so if we look at that 2% Phytophthora species, all of the Phytophthora species that we recovered from propagation systems were Cinnamomi. As soon as we moved to the container system, that 40% Phytophthora that we're isolating there, this is the species breakdown we saw there. Mainly Phytophthora pleura. That's a big change, and about a quarter of what we recovered from container systems were Cinnamomi. Compare that now to the field, where suddenly we have 10 different Phytophthora species. Again, Pleurivera is still slightly more commonly encountered than Cinnamomi, but you have this whole slew of other species coming in. And what we're seeing is, if you think back to the damage slide back here, we're seeing a correlation between the amount of Phytophthora that we're detecting and the number of species we're detecting and damage. So I'm not going to say too much about the Pythium species. We found on average 10 Pythium species from each of these systems. The most common Pythium that we discovered at all nurseries was Pythium cryptoirregulare. That leads to the second question we had is, how much damage do these pathogens cause? We took the 5 most common species that we encountered across all of the nurseries. Pythium cryptoirregulare was number 1. Followed closely by P. pleurivera and Phytophthora cinnamomi. Those were at most nurseries. And we inoculated rhododendrons with those. And what we found is that Phytophthora cinnamomi, pleurivera, and pini consistently kill, on average, 80 to 90% of the plants that we inoculated. Cryptogaea was a little bit weaker, killing about 35%. Pythium cryptoirregulare, the one where we found most often, this was the number 1 pathogen we found everywhere, only killed 3 plants in the entire study, and that is on par with the number of plants that died in control treatments that had no pathogen at all. And the reason for that is that these slightly larger plants that we were inoculating, these were about 4-inch tall plugs. They had already developed sort of a woody root system. They were big enough that Pythium really didn't have much of an impact. We really only see Pythium causing damage on really young seedlings that don't have much of a root system. We don't see much damage at all by Pythium once those plants become larger and have developed an adequate root system. One real quick thing, we did look at fungicide control. Growers have 2 main ways that they apply these 2 most popular fungicides. One, they will either apply it as a foliar spray to try to control above ground, like Ramorum blight and other foliar ones, and they're hoping that systemic activity will carry down to the roots. Or they can apply fungicides as a soil drench or a granular. And what we found is that the soil drench worked better than foliar applications for Cinnamomi. You see that over on the left where the foliar applications of both fungicides, we still had 80% mortality. Mortality was dropped by half or more once we went to soil drenches. But for Phytophthora pleurivera, nothing worked. We basically had 60 to 80% mortality regardless of which fungicide we used and regardless of where that was applied. So that made me think maybe we have fungicide resistance going on. So we did some analyses of that, and we tested all 35 isolates of Cinnamomi that we had from the nursery industry. And this is just showing a rough measure of how well each of those 35 isolates grow at different fungicide concentrations on the left. 40 is the maximum label rate of this fungicide that can be applied. And all of them didn't grow much above 1 microgram per mil of active ingredient. So this maximum rate is more than enough to control all of the cinnamomum isolates that we tested. Compare that to what happened for P. plurivera. We tested 53 isolates there, and there's 40 line, that's the maximum label right there. This is the scale here. We found many isolates of Phytophthora plurivera that were resistant to mefenoxam. And the same thing held true with phosphorus acid, and I'm not going to show you that here. But this is where it's really important to know which pathogens you're dealing with, because Phytophthora plurivera has this propensity to develop fungicide resistance. And that is probably one of the main reasons why root rot control is still poor in the ornamental nursery industry. So the take-home points that I really want you to go home with is sometimes growers are treating the wrong part of the plant. By applying the fungicides to the foliage, you're not necessarily going to get good control of root rot pathogens down in the soil. There's just not enough of that product going down into the root system to provide good prevention there. Second take-home point is the treatments designed to control Phytophthora cinnamomum are sometimes less effective against Phytophthora plurivera. We're starting to develop more things showing that all of these Phytophthora species are acting very differently. They have different susceptibilities, and treatments that have been designed historically for one species aren't necessarily going to work for other species. And so you really need some sort of broad spectrum control that's going to work against multiple species. And the last thing I know, I heard that this is a group that is really all about zero tolerance. And I think this echoes a lot of what you've heard throughout. If you have zero tolerance, my #1 recommendation to you is don't grow your plants in field soil. Grow them in containers, use clean pots and clean potting media, because the #1 way, the most damage that we saw was when there is that contact with field soil. And with that, thank you very much, and I'll open it up to questions.
Thanks so much, Jerry. That was a really interesting presentation. And your last point is one that we definitely are very aware of and concerned about here in California. Is in anytime we have nursery plants on the ground and in contact with soil, we definitely see issues with that, with pathogen contamination. So that's an important point to make. So Ted Swicky had a question earlier in your talk. He was wondering, for the identification of root rot, is that based on visual symptoms, or did you do isolation or baiting techniques for that data set?
This was all based on visual symptoms. So we were looking for symptoms of wilting, chlorosis, stunting, and we didn't find much of that in the propagation system. So there we were primarily sampling healthy plants, just trying to find pathogens. And Phytophthora only came from the few plants that were wilting there, but all of the rest were showing severe symptoms of root rot, and we would isolate from those.
Great, thanks for that. Then we have a couple comments from Tyler. Tyler says the Globisporangium pythium cryptoirregular species complex is the most common pythium we have in outplantings in California. And then he makes the comment that Phytophthora plurivera is not common in California, and we should be very worried about it.
Plurivera, so yes, we definitely find cryptoirregulari everywhere. It was one of the most common ones in forest nurseries here on seedlings. Plurivera is more worrying to me because it has an above ground component. It's also one of the most common species based on Jennifer Park and Nick Grunwald's work, where they looked at above ground infection on rhododendrons and many other species. And because it has that above ground component, I suspect why it's become more common over time. It makes it easier to spread because it has that above ground component, and it can also go below ground. I wonder if you're not seeing it in California. I mean, I would have expected that you would be finding it. And the fact that you're not finding a lot of it makes me wonder about host associations, and maybe you don't have the right hosts or the right environment for it. But I'm not sure.
Susan Frankel's asking, what percentage of Pacific Northwest rhododendron growers use containers versus field-based soil?
Ooh, that's a good question. I would say maybe we're looking at 60% are in container, but that is a very rough guess. I want to say 66% of the industry up here is roughly container, but I'm not completely sure.
And then Sarah Green is wondering, Phytophthora pini, it's not a species that she finds in UK nurseries. She's wondering if it's particularly common in California and on which hosts generally. And I'm not sure if that's a specific question to you, Jerry, since you're based out of Corvallis, but maybe if someone has a response to that, they could put it in the chat. Although actually Tyler just says second most common member of the P. citricola species complex. Sorry, guys, I'm catching up here on the chat.
Yeah. And I do find a lot of that. I've looked at a lot of survey work, and pini does come up quite a bit. I know it was real big in New York when we were working on cankers of European beech trees, of historic European beech, and pini would pop up quite a bit there, as well as plurivera.
And I think we'll do one last question before we move on to our next speaker, and we can take the rest of the questions at the end today. So Joanna Del Castillo is asking if you've tested pathogenicity of other Pythium species recovered.
We have, and they have a hard time. Our second most common one, I think, was macro, oh shoot, I'm not going to remember. And we have a hard time even seeing a 5% effect on overall plant size with the size rhododendrons that we're dealing with. I suspect it might be more of a rooting issue or seedling issue, just as those roots are forming and they're really young. But we don't see much of an effect of Pythium on plant health of the size of plants that we're dealing with.
Yeah, that's interesting, because I know this group is very concerned about Phytophthora, but Pythium is definitely another pathogen that I think ideally we don't want in our restoration outplantings, certainly.
It's a difficult one to get rid of. It seems more prevalent in a way, and I just don't see much damage caused by Pythium species unless it's with smaller plants.
Mm-hmm. Great. Well, thanks so much, Jerry, for joining us today. So our next speaker is Valerie Aviner. She's a professor and ecologist in the Department of Plant Sciences at UC Davis. And she's been conducting research to improve restoration outcomes for several years, focusing primarily on soils. She's a fellow of several societies, including the American Association for the Advancement of Science. And she's also an outstanding mentor for students and works to inspire urban youth to work in environmental science. She uses an integrative research approach in collaboration with a variety of different land managers. So thanks very much, Val, for joining us today.
Thank you all. And can you see my slides all right? Somehow I'm not seeing what's projected.
We do.
Great. So thanks so much for having me be part of this session. I'm already learning a lot, and I want to first acknowledge lots of other folks who've been involved in the study, particularly in some of the Phytophthora aspects. And so when thinking about restoration success and failure in California's riparian areas, I started talking with Mia and Golia about this a while ago, and being the soil nerd that I am, we were thinking a lot about soil conditions that limit riparian seedling survival. And big laundry list of soil conditions that really can influence what resources a seedling has, what kind of stresses that it experiences. And when she and I started talking about Phytophthora, I went through the literature to look through a number of soil conditions, because of course the issue with Phytophthora is once we have it in a soil, it's very hard to get rid of. Even, orchards that can do very intensive soil treatments really have found they've never successfully gotten rid of it. It's still sitting there kind of hiding in the soil and can pop out during wet years or things like that. And so a lot of these more intensive management systems have gone to ameliorating soil conditions as a way that Phytophthora is still there, but maybe it decreases its impact on plants. And I guess not surprisingly, but it was surprising at the time, it's the same list of soil conditions, right? So things that influence, how much, it's a water mold. So how much water is in, the soil, how well can water come through, but then also drain out. But also if you're a plant that's got a compromised root system, if you have a very droughty soil, you're going to have a tough time. So, in the wet times, you want good sort of water passing through and not pooling, but in the dry parts of the year here in California, you want good water holding capacity, in that soil so that it's not an added stress to a plant that's already got compromised roots. So things like higher water holding capacity, which might be enhanced by organic matter and things like that. And so our question became, can restoration success be improved by targeting activities on promising soils? So just picking what the most promising sites are and or restoring soil conditions before planting or seedlings. And so there are two main types of things we did. The first is looking at seedling health and how it's associated with soil conditions. And this was done in two different ways in two different sampling periods. And so in the first, in 2019, we actually looked at individual seedlings. And so you can see on your right, 3 sycamore seedlings that are right next to each other. The one in the middle is gorgeous and super lush. The one on the left is kind of hanging in there, and then the one on the right is dead. So we looked at ones that were nearby each other to see how soil conditions may have varied in relation to health status. Then in this last year, we did a similar thing, but we actually looked at broad areas of the floodplains that had either high survival, which was 50 to 100% survival of tree seedlings, high mortality, which is only 0 to 6% survival of seedlings, or real hodgepodge of a mix of live and dead seedlings, so about 20 to 50% survival. And on these plots, we measured seedling, obviously survival, but also things like height and canopy diameter. We looked at where that seedling was in the floodplain, so how far away from the creek it was, both in terms of horizontal, but also vertical, like how far away from the water, the groundwater it might be. And looked at a whole suite of soil conditions, not just in the surface soil, but deep down as well, because for a lot of seedlings, especially getting through our rough California summers, getting their roots down into some water can be really important. And then, of course, Phytophthora, which is the key point of this meeting, we looked at in 2019 in collaboration with the Rizzo lab, doing parabaiting for Phytophthora on our soil samples. And then in this most recent round, we're using DNA analysis, but we're mainly looking here at bacterial and fungal communities, including mycorrhizae. And so for this latest round, everything's still kind of slow with COVID. So some of the labs, such as the microbial community, that does the microbial community analysis are running behind. So we don't have those data yet. So that was looking at seedling health. We also looked specifically at Phytophthora and to what extent soil conditions influence the presence and potential impact of Phytophthora. And so across the sites that we worked at in the Bay Area of California, they'd been extensively monitored in the past for many different Phytophthora. And so we looked across lots of different sampling points that had been previously assessed and revisited them to see in areas that didn't have Phytophthora before and that did have some of our most noxious Phytophthora. And so here are maps of 2 of the sites. Like all the white points are the ones that we measured. The ones that are circled in blue are the ones that we got positive Phytophthora for. And so it was essentially very low detection of Phytophthora. We expected a lot more than this, and there can be 2 reasons for that. One is it was a pretty dry year, and I know in last year's session of this conference, a lot of people were talking about dry conditions are pretty bad for detecting Phytophthora, even if you are saturating the soil afterwards. And then also we were testing the soil, which is less reliable than testing the plants themselves. So we didn't see a strong relationship between current and previous measures of Phytophthora, in certain planting basins, but that again may have been our methods. In terms of if Phytophthora is influenced by soil conditions, we only had 2 sites that had really any substantial amount of Phytophthora detections that we could even try to do any statistics on. And so at our wetter site, we had 7 positive detections across 34 seedlings we measured. And what we found, so you can see on these graphs, seedlings that had no Phytophthora detection are labeled none on the left, ones that had Phytophthora are on the right. And what we saw is areas that had Phytophthora had lower soil organic matter, lower soil carbon, and lower soil nitrogen, all in the deep soil. And again, we know for riparian seedling survival that deep soil conditions can be really important. Looking at a drier site, we measured a lot more seedlings. We had 110 seedlings that we measured the soil associated with them, and only got 15 Phytophthora detections. And in this case, what we saw was Phytophthora presence is related to, again, the detected ones are on the right of each. So there's lower soil organic matter deep in the soil, same as at our drier site. But then what we also saw, as our wetter site, but then what we also saw is, not surprisingly, areas with higher soil moisture, higher soil compaction, which can prevent water from draining, and higher salinity had more Phytophthora. So our study on Phytophthora was actually pretty minimal because we didn't find that many Phytophthora. And so we were also going to link that with plant health status and plant performance, but we didn't have enough replicates to reliably do that. But from the limited insights we do have, highlight what's already been found in the literature, which is higher soil organic matter, higher infiltration, but also leaching can be really important in terms of minimizing the impact of Phytophthora. And so here there can be a couple different things going on with the higher organic matter. We do see a lot more activity of beneficial microbes that have been shown to compete with Phytophthora, and also things like mycorrhizae, which can limit Phytophthora association with roots. And then also in terms of the water infiltration and the drainage, it's 2 pieces, right? One is in the wet season, we don't want that water to pond. The other is in the dry season, we want enough water that's being actually stored in the soil to keep the plant with minimum drought stress. So moving to looking at seedling performance, again, as I mentioned, we did this on 2 different scales, one looking at individual seedlings that were nearby each other, and others looking at larger scale plantings that were successful or had poor establishment. And so I'm going to walk through a few different species because a lot of those effects are very species specific. For sycamore, to start off with, first we looked at what the soils look like under mature stands compared to in these restoration areas. So like how off are our soils as places that might be able to support these seedlings. And so right here you can see these nice giant beautiful sycamore in the areas that seedlings were planted. And this is just one graph of the example of the things that I've listed here, looking at surface soil organic matter, and these are live and dead seedlings of what got planted, and then compared to soil under the mature sycamores. And you can see under the mature sycamores, there's 2 times higher soil organic matter, 40% higher water holding capacity, as well as cation exchange capacity, as well as calcium and boron, which are things that have been shown to influence Phytophthora. and they're deeper and moister soils. And so we already may have challenging conditions because, right, seedlings that are dropping right under those mature sycamore in that much healthier soil may have an advantage compared to these restored sites where the soil is a lot poorer. When we think about how these soils influence sycamore, is a little bit site dependent. So first site is a very shallow drainage. It winds up being seasonally saturated. And so both when we look at individual seedlings in 2019 versus broad areas of success and failure, we found the same kind of things, which is that things that alleviate soil saturation are likely to help sycamores. And so seedling size increases when there's lower soil moisture, when there's lower clay, when there's lower organic matter, and lower water holding capacity. In contrast, when we're in a site that's a much broader floodplain and that maybe only has some very seasonal flooding and tends to be water limiting to seedlings, we did see that seedling survival and growth increase with the opposite, with higher water holding capacity, with higher clay and silt, and with lower sand. And right, so when we're thinking about the soil conditions that are going to be best for seedlings, we have to think specifically about what's limiting the plant. So is it that the soil is saturated, right? And or is it that water is limiting in that site? When we look at coast live oak, again, we start by looking at soil under mature oaks compared to nearby seedlings that are planted in the restored areas. And here we didn't actually see a difference in any soil variables under the mature oaks compared to in the restored sites. So that's good news in terms of maybe less soil amelioration is needed in terms of restoration.
Val, you've got 2 minutes left.
Okay, awesome, thanks. So when we're thinking about how soil conditions relate to seedling size and survival, again, it's going to depend on the site and whether it's a water limited site or whether it's a water saturated site. And so same things that we're thinking about, soil organic matter, water holding capacity, soil moisture are going to be really important, but you have to adjust that depending on what the conditions of the site are. Similar in terms of willow, that there's the same suite of soil conditions can be very important in terms of limiting seedling growth. And so just to quickly sum up then, soils definitely are impacting where we saw Phytophthora across the landscape, as well as seedling health and size of our riparian plantings. And so this can lead to some guidelines for selecting promising sites for restoration. So right, in wet sites, we're going to want areas that minimize water saturation, so places with lower clay and lower soil compaction. In dry sites that have water limitation, we're going to want higher clay and higher organic matter and water holding capacity, especially in those deeper soils that hold the moisture during the dry summers. And as with areas with Phytophthora, again, it looks like areas with higher deep soil organic matter and nutrients and lower compaction are going to have less detection of Phytophthora. And this also then leads to potential use of amendments to ameliorate soil conditions before planting. So things like adding soil carbon in various forms, especially getting those deep into the soil layers, as well as adding beneficial microbes such as mycorrhizae, which may be able to infect the plant roots and decrease contact points for Phytophthora. And my grad student, David Mitchell, is working with Point Blue to look at some of these practices in restoration. So thank you so much, and I'm happy to take questions.
Thanks, Val. That was so interesting, especially when we're thinking of riparian sites that have had altered hydrology where you don't have maybe an ephemeral creek, now you have a perennial system, and how that affects the native plants, riparian trees that are adapted to grow in one water regime or the other. Seems like it just complicates things even more for us, really.
Yeah, I think the whole aspect of like native restoration in riparian areas is so hard because the hydrology, one way or another, is so messed up that I think we have to think about maybe sort of moving which native plants might be in which watersheds, just because of what the hydrology actually is now.
Yeah, extremely complicated, but really interesting work. I have one question so far from Ted. Ted is asking if there's a relationship between the 0 to 30 centimeter organic matter level with Phytophthora, or whether it was only the deeper organic matter.
Really good question. It was much stronger in the deeper soil. There was a weak relationship in the shallower soil depths. So like we did 0 to 15 and 15 to 30, and it got basically each layer you got deeper, it got to a stronger relationship.
And Diana Benner is asking if you have any thoughts on care in using mycorrhizae inoculants, especially the commercially available products.
Really good question. In terms of the commercially available, right, I think when you've got a mine site or something that's completely devastated, sure, why not? But I think in more of these, altered but still, semi-functioning natural systems, I am not a proponent of commercially available, more sort of local, taking local whole soil inoculum and using those in these kind of areas. So of course, right, one of the things you have to be careful about is you're not moving Phytophthora around the, the system. But so for example, like those sycamores that had such different soil properties under the mature, sycamores versus 2-3 meters away where they're, where the restored plantings were, those kind of local movements could be really promising in terms of that inoculation. And again, hopefully my grad student, David Mitchell, will be able to tell us more about that in a few years.
Great, thanks again, Val. So that kind of, speaking of sycamores, is a segue into our next presenter and her talk. So we are going to hear from Carrie Frangioso, who is in the UC Davis Rizzo Lab, where she's worked for the last 20 years on sudden oak death and other forest health concerns along the California coast. She lives and works out of Big Sur. Today, she's going to introduce us to a relatively new research project looking at California sycamore health. And I'll also just mention that Carrie is also a first responder and firefighter for the Mid-Coast Volunteer Fire Brigade. So thanks very much, Carrie, for joining us today.
Here. Can you see my screen?
We see you.
Oh, here we go. Here we go.
There we go. Yes, thank you.
And you see the presenter view? Oh, here we go. Okay. Okay, so thank you, Susan and Janet, for asking me to come today. Thank you so much, Valerie, too. It is a really great segue into the work that that Tyler, Dave, and I did on the San Francisco Public Utilities Commission land this past spring. As we all have heard, the health of California sycamore is a concern across its range. It's mainly found in the riparian area, and there's a whole suite of abiotic influences such as grazing and hydrological alterations and all the things that Valerie just talked about, as well as biotic influences such as pathogens, drought, climate change, and the pond contamination from the introduced and widely planted London plane tree, which has really reduced the amount of suitable habitat for the species, which is estimated to be less than 5% of pre-settlement areas. So a major effort has been undertaken. I recognize a bunch of the pictures from Valerie taken in the San Francisco Public Utilities area of Sonol. They have made this enormous effort to try to restore this riparian area. They've planted nursery origin sycamores yielding a relatively low success rate. Sounds like, Valerie, your lab is really underway in trying to figure out what is causing the success and not success. Our goal was really to try to understand how that work has impacted the sycamores that have been there for very many years, more the mature sycamores that are there. And unfortunately, this restoration effort has introduced a whole suite of soil-borne phytophthora. So in the early spring of 2022, we at the Rizzo Lab established a long-term monitoring plot to monitor the health of sycamores in the Sunol area and to see really, is there a big difference between the restoration and non-restorative areas because of the introduction of these Phytophthora? Are the sycamores there less healthy? Are they dying more often, more frequently? So what we did was we set up quarter-hectare plots, pretty large plots, so that we could get some trees in there because they're pretty far spread apart. We measured all stems greater than 1 centimeter diameter. Multiple stems make up individual trees. Each tree got a numerical health rating, 1 being healthiest and 5 being dead. Attributes like basal sprouting, heart rot, and anthracnose and evidence of other pathogens were recorded on a tree basis. We also did a survey on the entire plot to see if we could find any naturally recruited seedlings, and they were then tallied by species. And then vegetation cover class of all woody plants was recorded. So we have 11 plots that we set up in non-restored areas and 12 plots in restored areas. Some of you may recognize these areas, these sites. This is a map showing where all of the previous Phytophthora have been found in our sites. This is a lot of work done by Valerie's lab as well as Phytosphere. And we pared down all of the Phytophthora that have been recovered in these areas to specific species of concern, and they involve Phytophthora cactorum, Cambivora, Megasperma, Tenticulata, and Crassimera. This is what one of the sites looks like. This is Sheep Camp Creek. It's the only site where Phytophthora cambivora was found. You can see Tenticulata at the very entrance of the area. But by and far large, the San Antonio Bioregional Habitat Restoration Area had the largest number of Phytophthora introductions into the restoration sites. Our plot locations were randomly located, irregardless of previous Phytophthora findings. There are non-restorative sites. This is the quarry site, Turner Dam, and Indian Creek. And so some results quickly, of the 153 sycamores that we found across 23 plots, 19 were totally dead. 3 only had living basal sprouts. The total number of stems was 366. 13% of those were dead. We had a large range of DBHs from 1 to over 200 centimeters. And the number of stems ranged from 1 to 18. We had 66 individuals in the non-restored areas and 88 in the restored, but that also includes 39 outplanted trees. And we did not find any naturally recruited sycamore seedlings or saplings throughout the entire survey. And that is probably one of the biggest concerns that we found from this study. And then to put this in more of a graphical way, you can see that nothing less than 10 centimeters is naturally recruited. Everything is outplanted, but otherwise you can see that the distribution of DBHs in both the restoration and non-restoration zones it's pretty well distributed. So of the 131 living sycamores, 56 were considered to be healthy and 16 were in poor health. Trunk cankers were observed on 48 of the living trees. The average health index across all sycamores is 2.15. So the lower the number, the healthier the tree. And when you compare the health index of non-restoration to the naturally recruited sycamores in the restoration area, it's 2.65 to 1.94. And the outplanted sycamores had the healthiest health index of 1.59. Of course, we're only looking at the any of the the planted sycamores that are greater than 1 centimeter and still attached to the ground. So regarding the sycamore fruit, 19% of the sycamores bore fruit in 2022. So it appears that quite a few of them bore fruit in 2022. And this is just 1 year of study, but 57% of the plots had at least 1 tree with fruit, and 45% of non-restored and 67% of the restored plots bore fruit. So in summary, we really observed few differences between the restored and non-restored sycamores, besides the presence of the smaller size class in the restored plots. Additionally, the San Francisco Public Utilities Commission have embarked upon a massive fencing project to exclude cattle from some of these areas. But again, this being just 1 year, time will really tell how effective these exclusions are helping with sycamore recovery. We found a lot of different pathogens: anthracnose, Massaria, powdery mildew, various insects and pests. We found various hot rot and canker-causing pathogens. But the 3 most important and common pathogens were the anthracnose and Massaria and heart rot fungus caused by Ionotus arizonicus. Sycamore anthracnose is caused by Apio pneumonia platani veneta. It's a complex disease. It has several different stages. When it's really severe, you can see complete defoliation of individuals. Typically, they send out a second flush of leaves after that. They do create these false witch's broom, which can interfere with fruiting. But this was nearly ubiquitous on every tree that we surveyed. The Massaria canker caused by the Macrodiploideopsis desmazeri was also nearly ubiquitous. We find these symptoms on exposed dead branches on most of the sycamores. It's really unclear at this point how Massaria and anthracnose might interact to stress or suppress the sycamore trees. And then pretty much every sycamore had heart rot. And this very characteristic is one of the most valuable ecological services mature sycamores provide. In most cases, in almost all cases, it was Ionotus arizonicus, which is a presumed native heart rot fungus pathogen. And it's worth mentioning that the London plane tree was introduced into the United States in the early 1900s. It's widely planted as an ornamental tree across the country and is considered naturalized in parts of California. The pollen from this hybrid can contaminate California sycamore populations, and additionally, it's probable that the anthracnose and Massaria canker are likely not native to the United States and were probably brought into the London plane tree with the London plane tree around the 1920s or 30s. So it's difficult to make conclusions after just having established these plots, but from our initial assessment, again, restoration efforts and the inadvertent introduction of various Phytophthora species doesn't seem to be impacting sycamore health. However, many questions still remain. Is recruitment happening elsewhere throughout the range of California sycamore? I open this up to the greater group to be a little more conscious when you're around sycamores. Do you see recruitment happening anywhere? And if so, what are the conditions that are favorable to that? And is it possible that the conditions needed for seedling recruitment might also encourage certain pathogens, which could be dormant and not doing much until those conditions occur? And they may then prevent the successful recruitment process. And then what are the long-term effects of various abiotic and biotic threats? I think, working with the Public Utilities Commission and everything that they're doing, they're really trying to get at that answer. And then another more theoretical question is, to what extent does a London plane tree, has it contaminated the California sycamore? And besides being resistant to anthracnose and Massaria and lacking the cavity forming properties important to wildlife, could the London plane tree provide some important ecological services? And that's it. I'll take questions.
Thanks so much, Carrie. Sycamore is such an important riparian tree. It's really, really great to have more research done on the species and sycamore alluvial woodland too, so rare in California. So thank you for that. We have a question from Charles McLean. He's asking, were the sycamore plantings propagated from seed or cuttings? Were any of the plantings hybrids of California sycamore and London plane? I'm not sure if that was part of your study or not, so maybe that's.
Yeah, I think Mia might be better equipped to answer that question.
So Mia, maybe if you want to put a response in the chat, since I think you're muted. And while you're doing that, I just have a comment. Carrie, you're asking about recruitment, what could potentially be limiting factors. So where I've seen sycamore recruitment is limited, and it's in areas where you're still having a natural hydro period and an unmodified creek, where you have ephemeral drainages. You don't see modified flow, which we get typically below our reservoirs. So in more natural hydro period areas where you still get that really strong channel forming flows and scour, and the longer period flood events that actually contribute to recruitment and regeneration of the floodplain. So when you can activate the floodplain and you have that more natural hydro period and then a summer drawdown but still high water table, that's where we're seeing sycamore recruitment, at least in Santa Clara County, on our native creeks above areas where we've changed the flow regime.
Great. I mean, this was just a huge learning process for us, and it's encouraging to hear that there is some natural recruitment happening, and hopefully we can see more of it.
Absolutely. Thanks again. That was a great presentation. So we are going to hear next from Mia. So maybe she has a chance to answer the question about sycamore plantings and propagation. So next we will hear from Mia Angolia, who is a senior biologist in the Natural Resources and Lands Management Division at San Francisco Public Utilities Commission, also known as SFPUC. And Mia works on many aspects of restoration and native plant nursery production for SFPUC. And she was formerly, she was a curator for the UC Davis Arboretum. And today she's going to talk to us about a project to produce mulch for restoration use on SFPUC managed land, which I know is a hot topic for all of us. Pun intended, possibly. And she's also a co-chair of our Phytophthora in Native Habitats working group. So thanks, Mia.
So there's a lot of sycamore questions. Should I just ignore them for now?
Yes, let's take those at the end.
Okay. Let me, hold on. Okay, that is not the right, see my title screen. I can't see you guys is the problem.
We can see your slide deck, but it's not in presenter mode.
Okay, let me try again. So when I do that, I can't see any of you. So I'm assuming you can see it. Is that correct? So if you're nodding your head, I can't see you is my point.
Yeah, that's correct. We can.
Okay. Okay. So I am going to talk about mulch today. First, I'll give a little introduction. So the San Francisco Public Utilities Commission, we provide water to San Francisco Public Services, wastewater treatment and disposal for the residents of San Francisco, and water through the Hetch Hetchy Regional Water System to 2.6 million customers in 4 Bay Area counties. I'm in the Natural Resources Division, and the Natural Resources and Lands Management Division is responsible for the stewardship of over 60,000 acres in 3 counties. These local watersheds are responsible for 15% of our water supply and most of our local storage. After witnessing the significant loss of tan oak and oak trees in the Peninsula watershed, followed by the introduction of multiple soil Phytophthora species that both Val and Carrie talked about in the last 2 talks, into our habitat restoration sites, we have banned the use of all outside container plants and have prohibited the import of most organic materials unless they're thoroughly heat treated to lethal temperatures for Phytophthora. One of our recent projects involves the landscaping needs of the Sunol Long-Term Improvement, which is a capital improvement project that's located on the edge of our Alameda watershed property, where we're improving and updating the facilities of an existing yard and enhancing the educational value of our Sunol Water Temple by constructing the Alameda Creek Watershed Center, an interpretive facility that will allow visitors to learn about the Alameda Creek watershed, its natural resources, history, and role in the SFPUC water system. The Watershed Center project includes a 3-acre native plant garden that consists of about 25,000 plantings that feature the plant species and plant communities of the local watershed. And the yard project included the planting of an additional 20,000 California native plants, mostly in bioswales that capture the surface runoff at the facility. There's about 100 species combined between the two landscapes.
Sorry, Mia, to interrupt you, but we're not actually seeing your slides advance. We're still on the first slide.
Okay, let's try again. I'll stop sharing and start sharing again. And I'll just do screen this time. Let's try that.
So I think now we're seeing slide 6, but it's, oh wait, let's see what's happening now.
Okay. Well, so this was the SFPUC overview. There's our watersheds. Do you see the 2 watersheds map?
Yes.
Okay. Here's the significant loss of tan oaks over the years. The intro to the Sunol Long-Term Improvement Project. This is the intro to the Watershed Center, and then the Watershed Center garden. And then this is the slide that I was talking on. So sorry about that, guys. Okay. So since these sites are wildland adjacent and we're going to have an ongoing need for plants, we had to determine how we were going to safely source all those plants and import large amounts of soil and mulch. For the soil, I found a 3-part planter mix that has 2 kinds of compost in it. And as the compost biodegrades, it actually generates heat and reaches lethal temperatures for Phytophthora. And after that treatment, it's handled and transported in a way that prevents potential contamination. For the container plants, we did construct a state-of-the-art nursery facility that adheres to the best management practices outlined by the Phytophthora in Native Habitats Working Group. So that's how we sourced all of those container plants. And for the mulch, we went 2 routes. At the yard, we went ahead and used rock mulch because it's very low risk and it's conducive to that because it's a bunch of bioswales. But for the center, I really wanted to use bark mulch. And so I made 3 attempts at creating low risk bark mulch since I couldn't find any suppliers that had heat treated mulch or anyone that could even tell me the source location of their material or exactly what tree species were even used to produce it. So during the first trial, we built a metal steam manifold that fits in the back of a 10 cubic yard truck, and we mulched the trees straight into the truck bed. We connected the manifold to our mobile boiler and covered the truck bed with a thick thermal tarp. We used metal thermometers to monitor different locations and depths in the truck and steamed the mulch for 45 minutes at 140 degrees Fahrenheit. The whole process of chipping the trees and the 45 minutes and everything actually took us about 4 hours. And since we needed just short of 1,500 cubic yards of mulch for the Watershed Center project, this meant that this steaming and mulching process would take us about 75 days. So that was way, way, way too expensive and way too long. So I went ahead and I did a second trial with the same manifold setup and this truck, where we attempted just to steam logs instead of the mulch, thinking that this might reduce the overall work time. But putting the logs in and out of the back of the truck actually was even slower than chipping. And the weight of the logs actually ended up breaking the manifold. And then it was like a week to get it fixed. And it was a whole thing. So it was at this point that I went back to the drawing board and I got Ted Zwicky on the phone, and we came up with a plan to produce some low risk mulch using material from the San Francisco from our peninsula watershed. So when large trees fail on our property, the trunks are taken to this log deck site, and then they're eventually hauled to the dump because we don't have a large trunk chipper to chip material of that size. So some of this material has been there for quite some time because we do what we can per year, but it's also very expensive to take it to the dump. So this top photo is an aerial photo of what we call the log deck. And working with the contractor on this we created a work area on the south side of the log deck, and we added an 8 to 10 inch thick layer of fresh gravel and created 3 working areas centered around these 8 by 10 foot metal plates that you see in the bottom left hand picture there. We required all the equipment to be cleaned and disinfected before being brought on site, and I even rejected some of the equipment that showed up with organic material in it. For log selection, we did not use any oaks, tan oaks, and bay because of pathogen risk. And we didn't use any cypress because they have a lot of nooks and crannies that could be full of dirt. And so they're very hard to clean. No logs that were touching the ground were used because they probably had more soil on them. And then they could have also had special status wildlife species under them. So we just wanted to let them sit on the ground. No fresh logs that had been cut less than a year ago were used, since they might still have active inoculum in them, and any tree with visible cankers was also rejected for use. The good news is that we still had plenty of large Doug firs and Monterey pines to pull out and to make into chips. We used a heel boom loader to pull the logs off the pile, being careful not to drag them, again, in case there was special status species underneath them. And then the logs were placed onto this first set of metal plates for debarking, where they were debarked by hand and then moved with the heel boom loader to the second set of plates. At the second work area, the logs were washed with hot water from a pressure washer on all sides, and the loader was washed down before being transferred to the third work area. The work plates were also washed down after each log was processed, and the plates were angled so that the wash water drained off the plates and into a channel where we created to send the water away from the work area. And most of those logs were, they were washed, every side multiple times before transferring them to the third area. And in the third area, the logs were split and then loaded into a trunk chipper, where they were chipped directly into a clean 35 cubic yard truck. All of the work areas were cleaned and sanitized at the end of each day, and the residual bark was hauled off to the dump. So when we sent a full truck to the site for chip delivery, we made sure that the trucks always stayed on paved surfaces and that their tires were disinfected when returning to the mulch work site. And in these aerials, you can see the progression of mulching and the mulch stockpiles, which are in that top right photo in the bottom left-hand corner of the top right photo. So this entire project, instead of that 75 days, only took 17 days, and we delivered a total of 38 trucks totaling 1,330 cubic yards of mulch. And so this last photo is an aerial of the garden, all freshly planted and freshly mulched. And while I feel good about this project and the mulch for this project, I do want to state that, this mulch is low risk, it's not no risk. And if you were to ask me if I was willing to do this in the heart of the watershed at one of our habitat restoration sites, I'm not sure I would say yes, even though I feel pretty good overall about the Watershed Center. In general, I think I'm just a bit gun shy after all of our experiences with introducing pathogens at our sites across the watersheds. Okay, that's it.
Thanks, Mia. I know mulch is a topic that a lot of us have been discussing for the past several years at least, and trying to find a clean source of mulch or a way to make our mulch clean. So Nikki has a comment. She feels like it might be best to only use mulch which is produced on a person's own property from trees there, and then keeping that material on site. Does anyone else have any thoughts or feedback?
Yeah, I just feel like this was like kind of the best we could, with the volume that we needed, this was the best case scenario and the best solution for that site, because every site's going to be different.
Yeah. So Diana's asking if you had a smaller scale project, perhaps the heat treating might be a more viable option, like if a much smaller scale.
For sure. It just for 50, we ended up only needing like 1350, but just it's a lot of mulch. for
Yeah.
Really big volume. But let's see.
Yeah, and storing it too is always an issue if it's not going to be used right away. Let's see, Ted is asking, how do you think the economics worked out? Your created mulch versus hauling logs to dump versus if you had actually bought mulch?
So I will say, for the same volume of rock mulch, the cost was about $200,000. And this ended up costing, it was about, I think, $240,000, $250,000, but it was also done through a change order with the contractor. And when we do a change order, there's like a much higher markup than if it would have been in the original contract. So that made it more expensive. And there was also like, there was the prime, the sub, and then another sub. So it was 3 layers of contractors. So, and every one of them charges us. So I think potentially if we had just done a contract for that, we could have maybe, gotten under 200,000 perhaps. So this mulch is specifically to the next question that says, what happens to the mulch next? So that mulch is specifically for that garden project. It's a 3.5 acre native plant garden. We required a 4 to 6 inch layer of bark mulch, and that's where it's getting used, and it will not go to the public. 4 to 6 inches, as Susan asked, what is the depth of the mulch on site?
Yes, clean sources of mulch. I knew Ted would have an interesting.
Yeah, so I mean, the thing, Ted is right about when they get hauled to the dump, we just have to pay it, and we don't use that anywhere. I will tell you that we pulled, it was 17 days and we pulled a lot of logs, and from certain angles of our log deck, you kind of can't even tell that any logs have been pulled because it's a really big log pile. So we're going to be pulling logs out of that pile for a long time. So I don't know, you say people insist that there are options to buy heat treated premium arbor mulch, but I've never found a clean source for mulch in which I feel okay. So I have called people. There was somebody that I found at one point that said what I needed was that if you went to mills, that there was like the leftover from milling and it's potentially heat treated, but I never found anything that actually like checked all the boxes and met all of our requirements.
Andrea is asking, has anyone looked at biochar under rock mulch as far as performance?
I have not. We do have a staff member who knows a lot about biochar, but I think it's probably something worth looking into, especially after all of Valerie's information.
Very great. Very cool, Mia. Thank you. This is definitely, I know, a topic that we'll all want to keep discussing, and as we're trying new things, and hopefully we have more options available. But it's always great to hear the work that SFQC is doing, because you guys do a lot of very innovative things. So thanks for that presentation. So our final presentation of the afternoon is actually 2 presentations. So first, we're going to hear from Diana Benner and then Christina Weber. So I'm going to introduce both speakers, and then they're each going to give their talks, and then we'll have questions for both of those speakers at the end of that. So first is Diana Benner, and she has a graduate degree in vegetation ecology. She's been involved in many aspects of native plant restoration over the years. Today, she's going to provide a short update on the Accreditation to Improve Restoration, or AIR program, that is in development for California. She's part of a small team that's worked very hard to set up an accreditation program for restoration nurseries. Diana is also a co-chair of the Phytophthora and Native Habitats Working Group, known as CalPhytos, and a co-owner of the Watershed Nursery and a leader of the Central Coast Native Nursery Network. Christina Weber is a senior environmental scientist and supervisor for the California Department of Food and Agriculture out of Sacramento. For several years, she's been working on the CDFA Nursery Best Management Practices Program with NORSDUC, National Ornamental Research Site at Dominican University. And the Best Management Practices Program is a voluntary program for both ornamental and native nurseries. So she'll give us a short update on that program. So we'll start with Diana. Please take it away.
Thank you. Hi. Hello, good afternoon, everybody. Let's see if I can get this to cooperate with me. And is that good?
Yes, thank you.
Thank you, Janelle. Okay, cool. All right. Hello, good afternoon. Yeah, I'm giving a short update on the Accreditation to Improve Restoration program. And I was really appreciating just coming back from the CNPS conference, the way many people were choosing to do some gratitudes up front. And I'd like to do that because there's a whole bunch of people who made this pilot phase possible over the past 4 years. And just super exciting talks this afternoon, just really quickly, and starting with that program in the UK is just awesome. Anyway, I have to give a huge shout out to Ted Sweeky with Phytosphere, who, you know, the Phytophthora and Native Habitat Working Group developed best management practices, but Ted took them and put them into this evaluation form, as well as being the lead in the team, the accrediting team. We're really excited to welcome Joanna Del Castillo back in the apocalyptic fall of 2020, when we were doing site visits with masks for both COVID and for fire fall issues. And of course, I have to mention Susan Frankel with the U.S. Forest Service Pacific PSW, who basically took the concept that we talked about in the working group and identified and implemented the pieces to make it a reality. And then that reality wouldn't have become a reality without the funders who supported the program. So SFPUC, National Fish and Wildlife Foundation, Santa Clara Valley Water, Mid-Pin Open Space District, and of course the awesome team at the CDFA Plant Pathology Lab, who are really amazing support in their diagnostic support for the program. And I basically wear the hat as sort of a nursery liaison and cheerleader in the process of implementing the rigorous BMPs of the program. And I just want to give a really quick, some people might not be familiar with the program. It was developed to support nurseries in their transition to implementing the CalPhytos, CNPS, BMPs, and to provide project managers the peace of mind that the plants they're using for their projects had been grown using rigorous phytosanitary BMPs. So the interested nurseries contact the team. They're given the evaluation form, which they fill out on their own, and then the AIR team visits the nursery and often conducts on-site testing at that time too, or in subsequent visits. And then the team and the participant work together to improve areas of production that pose a risk for Phytophthora infection. That was a really brief description, but that I think encapsulates it. So the principle of the AIR BMPs is based on exclusion of Phytophthora. So start clean, stay clean. And we're super excited to report that after 4 years of testing, so 564 leachate baiting tests conducted over those years. And I think the number is actually over 20,000 actual plants now. I think it's closer to 25. Ted can weigh in with that. 0 Phytophthora detected. So that's like, oh, it's working. Very excited about that, especially after all the work that goes into it. And that's compared to the amount of detections in nurseries where there's partial compliance, that we're closer to around 25% detection, and nurseries that were not implementing the BMPs, where about 71% of tests were detecting Phytophthora presence. So a really big impact that we've had in that amount of time. Very exciting that the comprehensive BMPs of both CalPhytos and CNPS can prevent Phytophthora establishment and spread in restoration nurseries. So this program is a voluntary program, and we really want to express our appreciation to the nurseries who have volunteered to participate. The first year of the program can be pretty daunting, but the program is structured to support nurseries during their transition. So they're not required to, or even really expected to have all the BMPs implemented prior to engaging in the program. It's a very wonderful thing I can talk about from my own experience of having support of expert pathologists in designing systems for the site. It's very nice. So currently we have 30 nurseries participating in Northern and Southern California, and about 7 are fully accredited now. I will say that because the BMPs are really extensive to implement, so far participation in the program has been a bit more palatable, I would say, to smaller nurseries. But other nurseries are also choosing to pick up an area of their nursery, a portion of the area, to be the AIR certified portion of their nursery. And I had mentioned previously Susan Frankel and how implemental she's been in manifesting this program. And she's really gonna retire soon. That's gonna happen. So we all had some trepidation around that. But thankfully, thanks to her efforts and many of the funders who have supported the program so far, it looks like the AIR program will be able to be housed at UC Davis in the plant pathology department. Previously, it was through the Forest Service Pacific Southwest Research Station. There we go. So that's really exciting. And we're still learning a lot and trying to streamline the program. And the exact configuration of how it's going to work with UC Davis position is still a little to be determined. And we're also exploring how to align with the CDFA nursery BMP program, which Christina is going to talk about next, to reduce confusion for nurseries and for project managers between the two programs. And I would say maybe fast, but I think that gets us pretty much up to date. And again, you know, a really big shout out to the AIR team, Ted and Joanna and Susan and all the funders, Cheryl and Suzanne at the pathology lab, and to all the nurseries who have so boldly agreed to take on this challenge. We're grateful. Signing out.
Thanks so much, Diana, for telling us about the AIR program and where it's at currently. So I think next we'll go to Christina Weber, who will tell us about the CDFA Voluntary Nurseries Program.
Hi there, everyone. I accidentally remuted myself partway through Diana's chat. So I'm Christina Weber. I work for California Department of Food and Agriculture. I think most of you are somewhat familiar with the CDFA NORSDUC BMP program. It's been around for some time now. I think it was maybe 10 or 15 years ago that they came up with the original manual that was geared specifically towards Phytophthora ramorum. So I didn't do a presentation because, you know, 5 minutes was not much time to get it in. So quick update on the program. Essentially, this is a risk assessment program performed by trained CDFA staff. There's no testing component at this time, but we are discussing potentially a baseline testing program. There's also no charge at this time, but there will eventually have to be a fee structure implemented. We're able to run right now based on being a pilot program and previous federal Farm Bill funding, and we're also pursuing other funding sources at this time. It is a tiered program, and we went to that probably in 2020 is when we conceptualized and started implementing a tiered program. What we found is that our BMP manual consists of 50 or so BMPs, including critical control points such as soil, water, plant buy-ins, things of that sort. And giving a nursery all 50 of those BMPs ended up being difficult to implement at once. So we came up with a tiered program, and what we found is we've had really good feedback from nurseries that it gives them something to work towards, and it encourages improvement in their best management practices to reduce pests and pathogens within a nursery. So this past spring, we performed audits at various nurseries throughout the state. That includes large commercial nurseries, tree nurseries, ornamental nurseries, things of that sort. And then we also performed audits at some of our participants that do native and restoration type outplantings and whatnot. There's several nurseries that have fallen within one of the tiers of the program, and then the rest of them are still working their way through and trying to hit one of the tiers of the program. And so we continuously work with them and provide feedback if they have questions and things like that. We have concluded our federal Farm Bill funding, and we've submitted all of our final reports to them. And some of the accomplishments for the season, we continued discussions with repeat Phytophthora ramorum nurseries, and we also work closely with NORSDUC to facilitate technology transfer from their research, and we help facilitate that so that nurseries can implement it. And we do that through hands-on experiences. So we go out to nurseries and we talk to them and work with them on some of the research that's been done at NORSDUC. And then we also do presentations to the industries as well as county ag commissioner inspectors. And then we have staff that will go to and give presentations at APS. Dr. Suzanne Rooney Latham, who I saw was on the call, she'll step in and give presentations and talk about the improvements for BMPs and all the great work that they're doing at their lab. I've given a few presentations with Diana at SoCal and California Forest Pest Council meetings and things of that sort. And then we also do outreach by participating in nursery trade events. So like the NorCal Landscape Show, or last year, we went down to Long Beach. And so we'll pair up with other CDFA folks that do our Don't Pack a Pest program. And we'll also talk about BMPs and things like that with people. And then we also have collaborated this year with SANC administrators to facilitate onboarding of that National Plant Board program at 2 nurseries in California. So we've been working to try to facilitate that program, as well as mirroring the CDFA NORSDUC program to the SANC program so that we can work together. And then let's see. We've also developed, so the Plant Pest Diagnostics Lab, Suzanne and Cheryl Bloomquist, they do a lot of great work with providing diagnostic services for the BMP program, NORSDUC, and native plant nurseries. And they also do a lot of work with pathogenicity experiments on potential new hosts for P. ramorum and other Phytophthora species. And yeah, I think the big takeaway that I wanted to bring to you guys at the table today with this program is that it is a voluntary program. I would say our focus has been originally on the large commercial shipping nurseries. And something that's been really cool lately is that we've actually been able to get a couple of them on board and done audits with them, and they They are in, we actually have a couple of them that are in our top tier of the program because they're doing BMPs at that high of a level. And of course, we're working with Diana and Susan to see how we can help out with the AIR program and where that might land.
Thanks so much, Christina. I'm curious if you could just give us a quick rundown on what the different tiers are in the CDFA program.
Yeah, so we're running the program on a 3-tiered system. So it's a gold, silver, bronze system. And so if someone falls within one of those 3 tiers, we consider them program participants. If they fall just outside of, say, the bronze tier, maybe they have a couple of BMPs left to reach that bronze tier, we consider them prospective participants, because again, I really want to be able to give those nurseries that are trying and they're implementing and they're continuously communicating with us. I want to give them credit where credit's due, because as you guys know, there are nurseries that have never seen what a BMP is in their whole lives. And I'm trying to bring those nurseries into the fold. And so we just revamped the tiers. So I don't remember exactly how many BMPs are in each one, but very basic BMPs in that bronze level, you know, checking your supplier, where are you getting plants from, you know, collecting plants in non-inclement weather. Things like that are in that very first tier of the system. Very basic, but very critical things. And then it moves up from there. So it builds upon itself. To be in the silver tier, you have to be doing every single BMP that is also in the bronze tier, and so on and so forth.
Thanks so much. So I'm going to open it up now to questions for either Diana or Christina. And we do have one from Andrea Williams. Andrea is asking Christina, can you talk about differences and similarities with SANC?
Yeah, so originally California was somewhat hesitant to jump on board with SANC because there's more to it than just implementing BMPs. A lot of it has to do with shipping and movement of plants between states. And so it wasn't until recently in Riverside County that there's a nursery down there that is highly interested in the program. And they, again, don't do testing. It's an audit-based program. And so similarity wise, that's what we are. We are an audit-based program. We don't do testing right now. So SANC kind of takes it, if you look at their manuals, they're much more in depth. Whereas our program, we have the basic BMP, like you should keep your hose nozzles off the ground, but we don't tell you how you should do that. Whereas SANC, that's something very basic, obviously, whereas SANC might prescribe it slightly more about how things should be done. And in general, I think nationally, that program has done quite well in Pennsylvania and some other states. It's just been strange. California has just not jumped totally on board with it yet. And some of it is just with, if you ship plants, you know that there's a lot of issues with quarantines and movement of plants because of that. And that's, I think, one of the reasons why California hasn't quite jumped on board with it.
Great, thank you. Are there any other questions for Diana and Christina?
I will put in the chat, we do have a website on CDFA's page, CDFA's website, that talks a little bit more. 5 minutes wasn't much, so it talks a little bit more about the program, you know, program documents, participants, things like that, and how to become a participant. And then also, if you're, the main thing for me is, yes, it'd be great for more participants, but even just people becoming exposed to BMPs is more important than anything. And so I encourage people, even if you know a nursery that doesn't want to become a part of any of these accreditation type programs, I would encourage them to at least take a look, because they might have something that sparks interest, or they're like, oh, that's something simple that I could start doing. So the website I just popped into the chat there.
Great, thanks. Yeah, that's a good point, that sometimes just starting with one simple thing that we can do in the nursery is going to be a huge help, and then we can build from there. So I'm not seeing any further questions at the moment. So
I think Diana is trying to unmute.
Oh,
Thank you. I'm sorry. I did the same thing. I muted myself and then I couldn't come back. I just wanted to say, because I see Nikki's question in there, and I wanted to give a shout out. Joanna and Ted are with the UC Davis staff are working on a website for the AIR program. So that will have participants listed there. But I also just want to encourage for consumers, like the more consumers that are asking, like every time you go to a nursery, any nursery, just start, you know, asking people about like, what are your best management practices for disease control? Because the more that gets out there in the public sort of norm, I think more nurseries are going to be realizing that it's a priority for people and interested in participating. That's my little plug. Thank you.
thanks for catching that question, Diana. Sorry, Nikki, I didn't see your question in the chat, but that is a great question. So I think it's good. We're all moving hopefully forward in the right direction. And the more nurseries that are accredited, the more options there are for all of us, whether it's restoration work or even people who want to plant in their garden, to know that we're planting clean plants. I think there were a lot of questions about sycamores and possibly mulch. So we have about 25 minutes left. We could go back to some of that discussion, or we could open it up if anyone has something specifically that they want to ask a question about or discuss that wasn't talked about, wasn't covered today. If anyone has a burning question, I think we're open to how people want to spend the last 25 minutes. Andrea, thanks for your comment. CNPS has started prompting people to ask about BMPs for higher risk plants. Yeah, that's also great. Just having more discussion, getting it out there, I think is a great direction for us to move in. So I'm also going to invite Janice to facilitate this part, because there was a lot of stuff in the chat that happened early on. So if we want to go back to some of those questions, we can do that. Or if anyone has a question that they feel wasn't answered, we could also ask that way, but I'm open.
I'm also going to we have about 40 people or so who are in this room. And so I'm going to trust we can all responsibly unmute ourselves to have a discussion. So I've given everybody the option now. If you would like to chime in, we can open this up to be a little bit less formal in terms of the presentations. We can go back up and kind of make sure we had a fair amount of discussion about sycamores. I just want to make sure if there was any lingering questions or comments about the sycamores, we could try to make sure those are covered and then go from there.
Janice, this is Faith.
Yeah.
Yeah. On the sycamores, I did get a reply to my question about the invasive shot hole borers, but it isn't only whether they're there now that I'm worried about. It's all this effort to try to restore sycamores, the shot hole borers are coming your way. They may not be there this year, but what are you doing to prepare for when they do come?
I mean, what am I supposed to be doing is my question.
Well, it is tricky, but the folks in Southern California have developed a lot of recommendations on how to manage at an early stage of the invasion. And so you should definitely be keeping up with that.
I mean, we do a lot of monitoring and we do a lot of, disease monitoring. And so having the Rizzo lab, having the Evener lab out on the ground, like I do think that early detection is key, and fingers crossed we've been good so far. But if I definitely, between Ted, Valerie, Dave, I feel like I lean on them a lot for advising us when we're in those situations. And those would be the first people that I would call to get advice on what to, and Susan actually. So.
Well, the people in Southern California are dealing with it now, and they're the ones who really know what is working and what isn't, as far as finding amplifier trees, getting rid of them, treating the others.
Just to quickly chime in, you know, we found all these pests and pathogens on these sycamores, but that was not the answer to all of the questions about the sycamore health by any means. And which was, you know, it was really huge project for, you know, just me as a plant pathologist. And so I think, you know, a more ecological approach like what Valerie Evener is doing is appropriate, because these questions of health are multi-dimensional, and there are a lot of, you know, there are multiple pathogens involved, there are multiple pests involved, but I think water and some other things along with it might be more important. Anyways, I'm gonna leave it at that.
Okay, it seems like maybe that's the end of just wrapping up the sycamore discussion. Unless I missed anything, feel free to let me know. I was looking back through the chat to see where we left the mulch conversation, and it feels like I think most people got their basic questions answered as best as we could. But if there's anybody who wanted to throw any last minute conversation in around mulch, I know it's a very important discussion, and if you had any lingering issues you wanted to bring up right now, we have time.
Janice, it's Susan.
Sorry,
Go ahead, Susan.
I have a question maybe for Ted. Is Ted still there? But the question would be, if I was using mulch, how would I test it?
Okay, if I've unmuted myself.
Yes.
Yeah, well, we run into this whole testing thing for large volumes of material kind of multiple times. We've just been working with people in NPS at GGNRA area on soil stockpiles, and it's always the same issue. Is you've got these huge bulk amounts of stuff and you wanted to find out whether it's contaminated. The problem is that these are usually not, they're heterogeneous materials. There's nothing to say that the whole stockpile's from the same source and all this other kind of stuff. So it really is a super testing kind of headache to try to say, it's sort of like it's actually very analogous to why the AIR program uses clean BMPs rather than saying, well, just test and find the clean material. The fact is, there's limitations to testing, and a lot of it has to do with the sampling. So what we mainly can do is look at the source. If you have some idea about, if you really have good provenance information about the sources, then you can start saying, okay, what's the risk profile of this? Is it high risk? Is it low risk? Is it no risk? So in a way, with Mia's material, the material that we were working with was off the ground. We were going to take all the bark off it. So we took all the soil that was off it. We're going to wash it down besides. So by the time we were done with that process, we essentially eliminated all the major risk factors associated with that wood. But if you're just sourcing it from somebody's yard somewhere where they've piled up mulch for God knows how long from God knows where, it's pretty hard to do testing adequately to say, yeah, this stuff is clean enough for us or not. So that's why you end up having to treat that kind of stuff. So long-winded answer, but it's kind of not, there's not really a good way to deal with it statistically, I don't think.
Okay, thank you.
I just wanted to actually scroll back to Charles' question. Charles McLean had a question about the sycamores, back to sycamores, and whether the hybrid with the London plane tree are thought to be less susceptible to anthracnose and whether that might translate to Phytophthora. I'm just curious of the pathologist's thought on that.
My suspicion would be those would be a different set of genes, but you never know. There's different types of resistance. There are types of plant resistance that are kind of more general. But it seems from my reading into it, although no one has really studied the genetic basis of plane tree to this anthracnose pathogen, but a foliar pathogen that's a true fungus versus the soil borne Phytophthora, which is a oomycete. And the fact that the resistance genes to the anthracnose can move, like they're a big part of what's driving the genetic pollution, is you have this invasive anthracnose that's been brought over from Europe, presumably, where the eastern plane tree Platanus orientalis is probably co-evolved with it. And so we have this pathogen, we have this hybrid plane tree that we're growing ornamentally, and the fact that the anthracnose is affecting the California sycamore makes the genetic pollution more likely, because the hybrids are more susceptible or more resistant to the anthracnose. So it's crazy, it's a double whammy, but I doubt that the resistance translates, although it definitely could. Okay, sorry about the long answer there.
I'll just toss in here that at least Phytophthora cinnamomum is known to affect London plane. So unless London plane has tremendous resistance to Phytophthora, there's no reason to expect that the hybrids would necessarily be more resistant.
Hi, this is Joanna. I have something else to add. Can you hear me?
Yeah.
Yeah, it's like I wonder also, like, what are the environmental factors that are favoring this anthracnose that we're seeing in sycamore? Say, like, is this favored by drought stress from the plants? And then if the pathogen came recently or it has been for a while, is the tree more susceptible for this fungus to become a pathogen? That's also something that would be interesting to see.
Okay, I was wondering if I could throw in a completely unrelated question. This is from a list of topics that have been kind of in the background. And I'm curious because we've heard from Val a little bit about the different aspects with soils, and a question had come in, not in this chat, but a different time, about whether healthy soils with appropriate levels of mycorrhizae have any natural resistance to pathogens, given that we talked a little bit about mycorrhizal additions and whether or not healthy soil should be moved in to provide a natural inoculum. I wonder if there's any kind of conversation we could have around the role of mycorrhizae in helping make soils less available for being, taken over by Phytophthora. If anybody has any insights on that.
I'm happy to chat if other folks, if there's not, anybody else with that, but I think there's two levels of it. One is mycorrhizae are never sort of only good, right? They can sometimes be, not pathogens, but they can be, net sucking from plants themselves. So it's not that mycorrhizae are always going to improve, plant performance, but in general, we tend to see that, particularly in resource limited conditions. And my understanding is particularly for ectomycorrhizal mycorrhizae, which essentially cover part of the root surface, it essentially almost physically inhibits other pathogens from getting, penetrating into the roots. And so it they provide some form of general resistance to pathogens that have to enter through the root tissue. There are other aspects of healthy soils, like there are a lot of, particularly in the rhizosphere, in that like 1 millimeter of soil around the roots that's super active, that there are bacteria and fungi that like actually can be active inhibitors to a lot of pathogens as well. And that's a little bit tougher in terms of like what the magic formula is of how to, implement that, because it's going to depend on the plant, it's going to depend on the soils. But in general, there's a feel from agricultural systems that when you have more of a healthy soil that has its own sort of maintenance of nutrient and carbon cycling, has a healthy and diverse microbial community, that those soils do have higher potential of suppressing pathogens, whether it's from, again, like the mycorrhizae from inhibiting entrance into the root, or by actively sort of suppressing the pathogens themselves.
Is that Valerie?
I'm going to make a couple of comments here. One, we have a lot of native systems that have been invaded by Phytophthora, and these are not native systems that are devoid of mycorrhizae and organic matter and things like that. We can have quite extensive destruction of habitats and host material in the presence of plenty of organic matter and mycorrhizal colonization. So by itself, that's not 100% protective. And then, again, it gets to the question about, well, can we, I mean, it may help, but there are such things as suppressive soils, and those things have been documented. They're not necessarily equally suppressive against all Phytophthora species, so that's another issue. But kind of a question back again, what I asked Val about earlier was that she mentioned this correlation with deep organic matter, but not seeing a strong correlation with shallow organic matter. But that's where most of the activity of the pathogen would be if we're going to talking about Phytophthora. So I kind of wonder whether, I mean, is that just a spurious correlation? One thing we have to remember on all these sites is that when you do a study at one site, it has a specific set of parameters associated with it, and not always are those things generalizable. I mean, the difference between the SAC site at SPUC, which was a heavily graded site, compared to the Adobe Gulch grassland site, which is not grazed, but just cut, wasn't graded. It was mainly the material was just cut down. Those are huge differences ecologically. So if you're making assumptions or generalizations based off of, these spots and then saying it applies to everything, I think you run the risk of really overgeneralizing things from small studies.
Well, it's making me kind of think about some of the sites that I work in that are naturally very nutrient poor soils. So kind of the question of what makes a healthy soil. So in serpentine soils, you're not going to have a lot of organic matter. You're not going to have nitrogen. You're not going to have a lot of the attributes that you might have in what we would consider a more traditionally healthy soil. And so it's just interesting when we're thinking of soil and potential soil defenses against pathogens. There's a whole variety of soils that are just naturally pretty harsh environments. And so how does that affect the whole pathogen invasion? And specifically, since we're talking about Phytophthora, it's just another interesting thing to consider.
Yeah, and I would agree with everything, Ted said, that there's not a silver bullet in terms of these ecological factors, right? There's just more of here are ways to maybe that are more promising, or less promising sites, or some other practices you could do that give things an extra leg up. I mean, as I mentioned in my talk, like what would the things that seem to be associated with Phytophthora or not, or with seedling success or not, changed depending on different site conditions, right? So it's not that there's, organic matter is always good, or something like that. And for Ted, for your question about the deep soil, my assumption there is that the deep soil organic matter, we've seen this a lot in grassland studies where we plant native grasses, and after about 10 years, the deep soils have far higher water holding capacity and organic matter in them. And what we're seeing is about 6 weeks longer growing season for all of the other plants in the area as well, just because it's keeping that system, right? It's keeping more moisture in that system over a longer period of time. So I assume, that kind of thing is more of an indirect effect where plants are probably less susceptible to, stresses in general when there's deeper soil water holding capacity and organic matter. And that can help actually fuel, say, more surface rhizosphere activity and things like that. But our study didn't go that deeply into it. In the latest version, we are looking more intensely at microbial community composition, but we don't have that data yet.
We only have about 5 minutes left, but there's a lot to be discussed here. I'm wondering if we could very quickly maybe just get to this other kind of question that leads off of this, which is what happens with the recommendations of steaming soils, in terms of killing off some other things that we might want to have available for plants when they're getting established. And there's been comments in the chat back and forth, so I encourage you guys to read through that. And if anybody wants to take a very quick stab at the role that the steaming of soil BMP plays in this connection to positive beneficial aspects in the soil.
I'd like to just jump in again on that, partly because we have to remember in the nursery, we're using soil-less media. I mean, they consist of forest products or peat and sand and lava rock and all this stuff. So we're not really dealing with a soil there that has a natural plant, microbial community. It's totally artificial system anyway. So what we're trying to do is make sure that artificial system, which has got really very little buffering in it, doesn't have Phytophthora in it as well. So that's where the steaming comes in. A lot of mycorrhizae will just blow it back in on the breeze, and you don't kill everything in there. There's going to be a lot of bacteria that will survive in those situations. But I guess my main point here is that nursery soil itself, which we're pasteurizing in the BMPs, is not some magic natural soil. It's this weird conglomeration of stuff that's made so it drains properly and can hold some nutrients and is aerated and works in pots and isn't too heavy, but it's not really a soil.
I think Nikki said it well in the chat, just to say that native soils are sure inspiring and complex. So I think this is one of these issues that's going to be continuing to be of interest to the group as we try to understand how to grow these plants as cleanly as possible and then get them established into a restoration area where they can really thrive with a native soil. I think with that, I'm going to say thank you. I want to thank all of the speakers today for spending your time, and we had such good participation with our questions and this discussion session at the end. So thank you to the speakers, the participants. Faith, yes.
Yes, sorry. We heard today about perhaps 3 different programs to try to ensure that nurseries are producing clean plants. SHANK is another one that we didn't go into in detail. There's a lot to be learned from the British experiment, I think. I'm not in a position to do this, but it seems to me it would be really valuable to have some Zoom calls or workshops of some way or other to compare and contrast and learn from each other and try to come up with something that everybody can endorse. Both something that's really good and something that, so there would be less confusion. Is that possible?
I know that we have been having long-term conversations between the AIR program and CDFA, which is why Diana and Christina presented together today. And those conversations about how the programs are similar and different from each other are still ongoing. I agree that we learned a lot from Sarah's talk earlier as well. I anticipate there's going to be more conversations amongst all of us, but we can definitely talk about making a more public educational kind of component where we can present them out to people. I know, again, Diana and Christina are doing that in person at things like the CIRCAL conference, but for other people to be able to participate, we can definitely look at doing something online. So thanks for that suggestion. We will definitely write it down and follow up. So just to wrap up my thank yous, I wanted to thank Janelle also for doing an excellent job moderating today, helping us with all of the speaker transitions and great intros and leading the discussions. It's now 3:30, so I'm going to sign off, stop the recording. I just wanted to let you know that the recording will be posted on the suddenoakdeath.org and calphytos.org website, hopefully in the coming week or so. Thank you so much for all of your time and participation today. Have a wonderful rest of your day.
Thanks, everyone.